Good day, everyone. Today, we're going to report the topic 6, absorption of fats, oxidation of fats, ketone, acetone, body ketosis. But first, I'm going to introduce to all of you the reporters, Danica Bonane, Junaya Bornalo, Francel Del Socorro, Sharolyn Pilayo, and Princess Sinahon. Absorption of lipids. Lipids are large molecules and generally are not water soluble. Like carbohydrates and protein, lipids are broken into small components for absorption. Since most of our digestive enzymes are water-based, how does the body break down the fat and make it available for the various functions that we must perform in the human body? So, it is from the mouth to the stomach. The first step in digestion of triacylglycerols and phospholipids begins in the mouth as lipids encounter saliva. Next, the physical action of chewing coupled with the action of emulsifiers enable the digestive enzymes to do their task. The enzyme lingual lipase, along with a small amount of phospholipid as an emulsifier, initiates the process of digestion. These actions cause the fats to become more accessible to the digestive enzymes. As a result, the fats become tiny droplets and separate from the water components. In the stomach, gastric lipase starts to break down triacylglycerols into diglycerides and fatty acids. Within 2 to 4 hours after eating a meal, roughly 30% of triacylglycerols are converted to diglycerides and fatty acids. The stomach churning and contractions help to disperse the fat molecules, while the diglycerides derived into process act as further emulsifiers. However, even all of the activity, there were very little fat digestion occurs in the stomach. The bulk of dietary lipid is neutral fat or triglyceride composed of glycerol backbone with each carbon linked to a fatty acid. Foodstuffs typically also contain phospholipids, sterols like cholesterol, and many minor lipids, including fat-soluble vitamins. Finally, small intestinal contents contains lipids from sloth epithelial cells and considerable cholesterol delivered in bile. In order for the triglyceride to be absorbed, two processes must occur. First, large aggregates of dietary triglyceride with a virtual insoluble in aqueous environment must be broken down physically and held in suspension, a process called emulsification. Second, triglyceride molecules must be enzymatically digested to yield monoglyceride and fatty acid, both of which are efficiently diffused or be transported into the enterocyte. The key players in these two transformations are bile acids and pancreatic lipase, both of which are mixed with chyme and act in the lumen of the small intestine. Bile acids are also necessary to solubilize other lipids, including cholesterol. Emulsification, hydrolysis, and micelle formation. Bile acids play their first critical role in lipid assimilation by promoting emulsification. As derivatives of cholesterol, bile acids have both hydrophilic and hydrophobic domains. On exposure to a large aggregate of triglyceride, the hydrophobic portion of bile acid intercalate into the lipid with the hydrophilic domain remaining at the surface. Such coating with the bile acid aids in breakdown of large aggregates of droplets into smaller and smaller droplets. Hydrolysis of triglyceride into monoglyceride and free fatty acid is accomplished predominantly by pancreatic lipase. The activity of this enzyme is to clip the fatty acid at positions 1 and 3 of the triglyceride, leaving 2 free fatty acid and a 2 monoglyceride. The drug Senecal that is promoted for treatment of obesity works by inhibiting pancreatic lipase, thereby reducing the digestion and absorption of fat in the small intestine. Lipase is a water-soluble enzyme, and with a little imagination, it's easy to understand why emulsification is necessary prelude to its efficient activity. Shortly after a meal, lipase is present within the small intestine in rather huge quantities, but can only act on the surface of triglyceride droplets. For a given volume of lipid, the smaller the droplet size, the greater the surface area, which means more lipase molecules can get to work. As monoglycerides and fatty acids are liberated through the actions of lipase, they retain their association with bile acids and complex with the other lipids to form structures called micelles. Micelles are essential small aggregates of mixed lipids and bile acids suspended with the ingest. As the ingest is mixed, micelles bump into the border of small intestinal enterocytes, and the lipids, including monoglycerides and fatty acids, are taken up into the epithelial cells. Absorption and transport into blood. The major product of lipid digestion, fatty acids and two monoglycerides, enter the enterocytes by simple diffusion across the plasma membrane. A considerable fraction of the fatty acids also enter the enterocytes via a specific fatty acids transporter protein in the membrane. Lipids are transported from the enterocytes into blood by a mechanism distinctly different from what we've seen from monosaccharides and amino acids. Once inside the enterocytes, fatty acids and monoglyceride are transported into endoplasmic reticulum, where they are used to synthesize triglyceride, beginning in the endoplasmic reticulum and continuing in the Golgi. Triglyceride is packaged with cholesterol, lipoproteins, and other lipids into particles called chylomicrons. Chylomicrons are extruded from the Golgi into exototic vesicles, which are transported to the basolateral aspect of enterocytes. The vesicle fuse with the plasma membrane and undergo cytosis, dumping the chylomicrons into the space outside the cells. Because chylomicrons are particles, virtually all steps in the pathway can be visualized using an electron microscope as the montage of image in the PPT. Transport of lipids into the circulation is also different from what occurs with sugars and amino acids. Instead of being absorbed directly into capillary blood, cyclomicrons are transported first into the lymphatic vessels that penetrates into each vellus called the central lacteal. Until recently, it was not understood how the large chylomicrons are taken up into the lacteals. As it turns out, there are patches of the lacteal in which endothelial cells are held together through specialized button junctions that are much more permeable to cyclomicrons than normal cellular junctions. Chylomicrons reach lymph, then drains into the lymphatic system, which rapidly flows into blood. Blood-borne cyclomicrons are rapidly disassembled and their constituent lipids utilized throughout the body. When large number of cyclomicrons are being absorbed, the lymph draining from the small intestine appears milky and the lymphatics are easy to see. In this picture of abdominal contents, the fine white lines are intestinal lymphatic packed with chylomicrons. That lymph passes through mesenteric lymph nodes and then into large lymphatics. Another lipid of importance that is absorbed in the small intestine is cholesterol. Cholesterol homeostasis results from the balance of cholesterol synthesis, absorption of dietary cholesterol, and elimination of cholesterol by exertion in bile. Years ago, it was shown that cholesterol, but not plant sterols, is really absorbed into the intestine. Most recently, a specific transport protein has been identified that ferries cholesterol from the intestinal lumen into the enterocytes From there, a bulk of cholesterol is esterified, incorporated into cytochromicrons, and shuttled into blood by the mechanism described in the picture. The next topic is oxidation of lipids, and it will be reported by Ms. Bornalo.